In the experiment for the viability of HL60 cells when exposed to mistletoe extracts, the goal was to find a vascular or non-vascular plant extract that can induce apoptosis onto HL60 cancerous cells. Research was done to find that mistletoe has been studied and found to be a great inducer of apoptosis in cancer cells. In fact, in Europe, mistletoe extract is among the most prescribed drugs for patients with cancer, but the FDA has yet to approve mistletoe as a treatment for cancer. Apoptosis, as you can see on the right, is a controlled cell death that happens naturally in all cells. It starts by the nucleus condensing and then fragmenting into smaller pieces, which get turned into smaller agents, which a macrophage can come and eat them up. Another cell death is necrosis, and necrosis is cell death by some outside factor damaging the cells. Necrosis causes the cells to swell up and then explode, causing all of the contents to be expelled out. To perform the experiment and collect the results, a viability assay was conducted. The viability assay shows how the cells can survive in the wells. A high viability shows that the cells are alive and thriving, while a lower viability means the cells are dying. For a successful experiment, you would want to get a lower viability assay with the extract, showing that the extract is working and killing the cells in the well. As we can see in Table 1, there are recording for the absorbance during the viability assay. The higher the viability, the higher the concentration of cells, and vice versa. The above table shows that the standard deviation and t-test control of our 70% ethanol and treated cell wells. The standard deviation shows how far the obtained numbers are from the standard. The t-test shows if the data collected is significant or not. If the value is smaller than 0.05, then there is a significant difference in the data. The control well has nothing added to the cells but the cells themselves. This will give the control number of cells that are not dying from any addition of material. The well with the 70% ethanol is used to ensure that when the cells are treated with the mistletoe extract, it is not only the ethanol that is killing the cells, but the extract that is killing the cells. The etoposide well has the cells treated with the mistletoe extract. This well is the well that can show the significant difference based on the extract works or not. Based on the results in the table, there shows a clear difference in absorbance between each control well and treated well. Both the standard deviation and t-test show that the treated cells show a significant difference in absorbance, as you can see here and here. Now the second table simply shows the average absorbance in each well of the treatment. Again, here you can see it is able to be confirmed that there's a large difference in absorbance or viability between the two controls compared to the treated. As you can see, there's two controls and then the treated cells. Now the chart below shows the average absorption but in a different view. The chart gives a better visual look on the significance of the treated cells compared to the control. And you can see here, signified by the star, that there is a significant difference between the two controls and the treated cells with the mistletoe extract. Another step in the experiment was conducting a morphology assay. And a morphology assay allows us to see the nuclear morphology of the HL60 cells after their treatment with the mistletoe extract. The morphology assay uses a DNA binding dye called cyto16 to see the degradation of the DNA through apoptosis. The cells were observed under a fluorescent microscope. This picture above shows the morphology assay of after 12 hours. Now there was a previous assay of 24 hours, but the extract's effectiveness was better than expected, with very little to no cells left. This picture is great to show the multiple steps of the apoptosis cycle cells undergo, such as cells you can see here labeled as with the red are cells that can be seen as healthy cells that have not yet undergone apoptosis. Now cells labeled with the yellow here and here, you can see that are beginning to start the apoptosis cycle with their DNA condensing and maybe even some fragmenting of their DNA. While there are also fragments as labeled in the blue of cells that have completed the apoptosis cycle and are floating in the medium as fragments waiting for a macrophage to come and eat it up. The picture is a great representation of the apoptosis cycle of the cells undergoing. Also, it can conclude that the mistletoe extract is a great inducer of apoptosis in HL60 cells. The last step of the experiment was to conduct a comet assay, and a comet assay is used to show the visualization of the damaged DNA in individual cells. The cell culture was again stained with the DNA binding dye Cyto16. Our cells were then placed in an agarose gel and ran through electrophoresis to show the separation of the cells and the damaged DNA. The picture shown here has the comet assay of our HL60 cells. The cells were able to be observed through another addition of the DNA dye, and then when it was dry, it went through multiple washes of TBE, chilled lysis solution, and alkaline solution. The gel also went through trips to the fridge and darkness to allow the dye to settle in the gel and also in the cells. After the electrophoresis of the gel, they were observed under the fluorescent microscope to see the distance of the DNA traveled in the specific cell. The cell above, as you can see right here, can be seen to have some diffusion of the DNA. Since there wasn't much diffusion, it can be concluded that the cell is still undergoing apoptosis because the DNA is still in larger fragments. The smaller the fragments, the farther away they would travel from the cell. As you can see, there's not much separation between the cell and the DNA. If we give the cell more time, you can see that there's a possibility that it could travel far, create a bigger tail on the cell. With the viability, morphology, and comet assay, it can be concluded that the mistletoe extract is a great inducer of apoptosis in HL60 cells. The viability assay shows the significance and viability between the control well and the treated cells, stating that the extract treatment was effective in inducing apoptosis in the HL60 cells. The morphology assay had to be done at 12 hours because the 24-hour assay had no cells to observe because the great effectiveness of the extract killing the cells through apoptosis. Even in the 12-hour assay, it can be seen that there are cells that were currently undergoing apoptosis and fragmenting, and also cells that have completed their apoptosis cycle and have already fragmented, waiting for a macrophage to come and eat them up. The comet assay showed that there was some degradation of the DNA in the cells of the treated extract, but it was too early to see some extremes in the degradation of the DNA, but can still show that there is some degradation. Now, this was an in vitro experiment, and there still needs to be some experiments with adequate results to transfer using mistletoe extract into clinical trials. If more research is done to ensure that there are good results that mistletoe extract can be used for patients battling cancer, then it is possible mistletoe can be used for patients as a treatment to kill their cancer cells without damaging other cells in their body. Now, I would like to thank you for taking your time on listening to my screencast and hope that the conclusions stated here can help me win the Nobel Prize on my mistletoe extraction's effectiveness on the HL60 cells. Thank you.